We end tonight with a mystery from the deep. Scientists on the West Coast are at a loss to explain what is killing sea stars, also known as starfish. Ben Tracy says in some places, 95% of the starfish population has died.
A sudden surge of the animals wasting away in just the past 2 weeks.
So-called melting sea stars were first noticed in Vancouver, then in Seattle, and now in California.
They are alarmed at a mystery epidemic.
What's been killing starfish?
At this time, we don't have a good idea of what's causing this.
Sea star wasting disease is a syndrome of a whole group of sea stars. So there's over 20 species in our waters that are affected by it. And what we see first is lesions appearing in the stars in their outer skin. And then those progress to eventually arms ripping off, and then the whole animal dies. The surprising thing in the initial outbreaks was how many different species were affected. So sea star wasting disease has been reported previously, but usually it's only in one small geographic area or just a single species of star. So it immediately got my attention to see 15 species kind of dying sequentially. And the striking thing about the sunflower star Pycnopodia is it was always the one that died first and most catastrophically. So we would call that a multi-host disease or a multi-host pathogen, and the sunflower star was the most susceptible of that. And so that's why we think it's becoming endangered. I just didn't really think this would happen. I did not think we were going to be listing an endangered species, 7 years into the outbreak, and here we are. We've just listed it 3 months ago as an endangered species. We've actually learned a lot about the ecology of some of these sea star species because they've disappeared. And of course, the ochre star was considered to be a keystone species by Bob Payne. I mean, he defined his whole ecological concept around the ecology of the big impact of that species. The sunflower star is subtidal, so we knew a little less about its ecology. But it turns out that removing it has caused urchins to explode in very high numbers, and they've eaten away their kelp beds. So there's a lot of concern, particularly in California, where the impacts are even more severe. Like there are no sunflower stars there. And there's huge explosions of urchins and decimation of kelp beds. So the ecological ramifications of this are kind of a big domino effect that is pretty serious, and it's affecting fisheries. It's affecting the crabs that live in the kelp beds, the abalone that live in the kelp beds. So it's a serious issue. And in fact, the sunflower star just this year has been listed with the International Union of Conservation of Nature as an endangered species. Well, one of the things we're finding is that infectious disease is increasing under warming conditions. And since we're expecting a lot more warming as our climate is changing, we can expect that there will be more new infectious diseases cropping up. And this is because both the hosts become stressed and the pathogens can cycle faster at warmer temperatures. So it's very important to be able to manage infectious disease better, both on land and in the sea. So the sea star epidemic is one of the kinds of things we can expect to see more of, just this sudden huge outbreak that correlates with warming conditions. And so it's very important to learn how to deal quickly and efficiently with these new infectious agents before they get more out of hand, and to learn better strategies. It's difficult in the oceans because these critters are out of sight and sometimes out of mind. And so we need better surveillance to pick these up as they're starting, so that we get the proper research so we learn better to control them. It's very hopeful the way the marine research community pulled together to study the outbreak. I mean, it's a huge, and citizens as well, it's a huge range that's affected from Mexico all the way to Alaska. And yet we really have pretty good data tracking the spread of the outbreak and the intensity of it. And similarly, we have a really good team of biologists now, and agencies like Nature Conservancy, Seattle Aquarium, really pulling together to try to devise strategies for how to manage a species that's impacted the way the sunflower star is, including, you know, development of a captive breeding program at a place like Friday Harbor Labs. And again, I think that's just such a good example of a kind of a crossover with research and a need, because Friday Harbor Labs is known internationally for its embryology research and its embryology courses. And so we were really well positioned to have a scientist like Dr. Jason Hodine, who could quite rapidly figure out how to breed. He's got baby stars that are the size of the palm of my hand now, that are a year old. And so that's a really impressive feat.
Yeah, my name is Jason Hodine. I'm a senior research scientist here at Friday Harbor Labs, and I study metamorphosis in the ocean of marine organisms. So we started in April 2019,
Okay.
and so a little bit over 2 years ago. It took until November until we got our first spawning of local stars. It takes time and patience and and a lot of attention, and they're hungry, and it takes a lot of food to feed them. Yeah. You know, people looked at sunflower stars that are this size and wondered, what is that? Is that a 5-year-old? Is that a 50-year-old? I mean, what is, and what's the minimum time, I guess, it would take to get to that big size? So we grow them for about 60 days through that first part of the life cycle in jars, and then they get to their settlement stage. We settle them out as little juveniles, which are super tiny. You can see them, but barely. They're about the size of a poppy seed. And then from that point on, they start to grow. If they find the right food and they're in the right conditions, they'll grow. And what we found is that they'll grow from that sort of poppy seed size to about the size of fitting inside the palm of your hand in about a year, which is far faster than we thought. We didn't think that we're going to get that big in a year. If they're this size in 1 year, and they're this size in 2 years, and that's reproductive size, we think they can reproduce in 2 years. Now, how long it takes to get from that to this, we don't know yet. But we think that in 4 or 5 years, you can have a full or mostly full-grown adult potentially. Thinking maybe next year actually as a potential, sort of first goal of maybe doing a small-scale reintroduction along with a monitoring project. So that would require additional funding. But the ideas of trying to reintroduce a major predator like this that's missing, I mean, that's a massive undertaking. We certainly don't think about what we're doing as sufficient at all. Our operation is way too small. If they're extinct or nearly extinct in the wild, it might not be possible to get local stars. But there are some things that you can do to check to make sure, I guess, that it might be reasonable to use stars from outside the region. And there are historical samples in museums, for example, that were collected all across their range, and you can get DNA out of those individuals. So they can do the studies to sort of identify what the population genetic structure is here, how it relates to the populations in other places, and to see, for example, whether or not the stars that were in California, are they really, really different or not from the ones here? And we actually have some insights in our work that suggest that we're guessing that there's probably a lot of what we call connectivity between populations. In other words, that the individuals move all along the coast, and that might be sort of I think most people, when they hear that, they think to themselves, wait a minute, a sea star is walking from Washington to California, and that's not what happens. These are quite mobile animals, and they have a large home range, but nothing like that, obviously. But it's during their larval stage, where they are floating in the plankton, that they could get carried hundreds or thousands of miles during their larval stage. And what we've noticed is that they have a potential of having an extremely long larval period. We are trying to do a lot of the things on a small scale that one could replicate on a larger scale too. So, we think about what we're doing is sort of putting together, a recipe book, if you will, for how to grow a sea star. And we want to, we're publishing that, and we hope that it's going to spur more people to do this, because the size of our operation is not big enough for the effort, but also just the challenges that we're facing in terms of optimizing this process. I mean, we're not like somehow uniquely super geniuses or something like that. I mean, it's just work. It just takes time, to like be able to, and exploration. The more people that we have working on this, the better. So we're trying to specifically clarify things that that anybody who puts the time and effort and has the basic skill set of raising larvae or doing aquaculture type work. We want to be able to promote them to do it. So wouldn't have been possible without the support of a place like Freddie Harbor Labs. I mean, we have this incredible recirculating seawater system here, which is why we're able to keep all of our individuals alive. We have 100 years of experience and research in raising marine embryos and larvae with all the embryo equipment and everything for being able to do this kind of stuff. We've got the greatest, the maintenance staff, that I know of, and the helping out, like building tanks and modifying equipment and so forth. And yeah, I mean, it's like, and as far as the broader community, it was really great during the time, especially I would say, especially during the time we were looking for stars in the first place. People were really great about letting us come onto their land to search for stars and send in observations when they thought they found a sunflower star. In some cases they didn't. They needed to send me a picture and there was a different kind of star or something like that. But in other cases, making sightings of them. I had a couple of crab fishermen send me photos of sunflower stars coming up in their crab pots, and these were really important in being able to identify what we've now found out are certain hot spots on the island and around the islands where the populations are still existing. They're not in every place where they were, just in reduced numbers. They're in specific locations, it seems, in reduced numbers, but gathered in those spots. The kayak groups were really keen on trying to find sunflower stars with their clients when they were out paddling around in various parts of the island, and that was really fun working with them.
It shares some similarities with our sea star pathogen in that it's COVID is a multi-host pathogen. It affects not only humans, but wild cats. It came from bats. It affects mink and maybe other things as well. So similarly, the sea star pathogen seems to be a multi-host pathogen. It affects over 20 different species of sea stars. And it's extremely transmissible and very fast spreading and extremely deadly. So again, our current count on the sunflower star is that over 98% of them are gone from the whole continental US. And then if you count all the way up to Alaska, it's about 90%. So in colder waters, you're surviving a little better. But they're really suffering hugely. Focusing on what is it that provides health for marine organisms will give us very valuable clues for also human health as well. And one big way back to a healthy ocean is more natural ecosystems and studying the pathogen fighting services of those natural ecosystems. There are a lot of ways forward. Obviously, reducing the pathogens we put in the water would be very good with better septic treatment. Very great care and best practices with aquaculture would be important.
Regardless of whether or not we have a direct consumption or something, if we see a direct benefit to humans. I mean, the benefits, are there, and they reveal themselves in kind of stark and shocking ways, really. I mean, and I don't want to say that that there's going to be a point in which we figured it all out, because it's never like that in science. I mean, there's always more interesting questions that comes.
We have at the Friday Harbor Labs a captive breeding program now that's been developed, and we're talking about how are we going to keep this animal, this endemic species, in the ocean.
So that was calamitous for many different species, but it was worst of all for the sunflower star. They were reduced in their populations, we think, by at least 90% or more in everywhere where they're found, just about, and their recovery has been, if at all, has been very slow. So that's those were the justifications for the listing. There was work in the 1960s, actually came out of the University of Washington. Bob Payne developed this idea of, well, he basically noticed that the predators in intertidal communities and in seafloor communities tended to have often incredibly profound effects on the structuring of that community. So when that predator was removed experimentally, or if it disappeared for whatever reason, that would then cause major shifts in the ecological community, and oftentimes reduction in the diversity of species out there. And that's actually exactly what we're seeing happen with the die-off of sunflower stars.